Good evening and welcome to Orkney International Science Festival 2021 online in this year of Scotland's coasts and waters. My name is Eric Walker and it's my pleasure to be hosting this final session of what has been a superb online science festival. Our speaker this evening is Dr. Annie Hill. Annie is Associate Professor and Programme Director for the MSc Postgrad Diploma in Brewing and Distilling by Distance Learning at Heriot-Watt University's International Centre for Brewing and Distilling. She has a background in microbiology and joined Heriot-Watt in 2001. Her research interests currently involve distilled spirits production with a focus on fermentation and also novel botanicals. She is co-founder of the Scottish Distillers Association and a member of the cross-party group on Scotch whisky. Annie surmises that it matters where whisky is made, but at the heart of the production process are the cells of yeast. What effect on flavour and aroma do these have? And could that give us clues to the fermentation environment of early types of Scotch whisky? Annie is going to describe her studies at places including Lindores Abbey, possibly the home of the first whisky production. Could I ask you all to please enter your comments and questions in YouTube's live chat, and I'll present them to Annie after her talk. Annie, over to you.
Thank you very much, Eric. Thank you for a kind introduction. And thanks very much to Howie and Louisa for inviting me along tonight. I feel very humbled looking at some of the speakers that have presented this week. It's I'm very honoured to be here and able to present some of our work. As Eric said, I've got a background in microbiology. So my sort of focus tonight is to champion yeast and to have a look at the impact that yeast has on whisky flavour, to describe the impact that it has and look at some of the studies that we've done on yeast and flavour and isolation of yeast from the environment, so including places such as Lindores, as Eric's mentioned. So just firstly to introduce where we are and at Heriot-Watt University. On the next slide, we have a picture of the building where we are. We're based at the John Muir building on the Edinburgh campus at Heriot-Watt. So we're on the western sort of fringes of Edinburgh, just on the sort of western side of the Edinburgh City bypass, if everyone knows where that is. We have campuses, of course, in Orkney as well, and in Malaysia and Dubai, and also in the Scottish borders as well. But we're based up in Edinburgh. And within our facilities, we have a range of different sort of pilot plants. We carry out work on the small scale. So the middle picture at the top there is a small alembic still, which is around about 1 or 2 litres, and we have these small stills that are up to about 5 litres made out of copper. We also do work with glass stills, but we generally do use those for gin work and things. We also have a pilot distillery, which is shown in on the right hand side. This is a 25 litre wash still and under 18 litre spirit still. So it's a double pot still distillation. And these stills were gifted to the university by North United Distillers. And they're made out of glass, but they have copper, sacrificial copper included in them. So we can alter the amount of copper that we have to replicate the same kind of copper content that you would have in a commercial operation. So we carry out a lot of our work across these stills. We have over 50 undergraduate students doing the BSc programme, and we have over 200 students that are studying our MSc programme. 70 of which over the last year were on campus, and then we've got over 150 that are studying by distance learning. So we have a very wide network and a lot of students and a lot of things that are going on. Below at the bottom of this slide, we've got a range of different companies that we sort of worked with on distilling projects. We've worked with both small and large scale companies. So including sort of Edinburgh Gin on the gin side, as well as Verdant in Dundee, Percy Distillery, that's in Perthshire, and Dunnet Bay Distillers. So they're all gin distilleries that we've worked with, as is Isle of Harris. We've also worked with rum distilleries, so Dark Matter, which is based near Aberdeen. With vodka, that's Ogilvy Vodka, which make a potato vodka. And then a range of different whisky distilleries as well. So that includes Port of Leith, which is being built in Edinburgh at the moment. The Three Stills, which is based in the Borders. The Oxford Artisan Distillery, and Lindores Abbey, as well as overseas companies such as Nika Distillery in Japan. So we have a wide range of different companies and different sort of scales that we work with on various sort of distilled spirits projects. But of course, today I'm going to focus on whisky production. So first of all, I'll go through a bit of the background and the process of making whisky. So on the next slide, we have a sort of schematic of the whisky production process. Many of you will be familiar with this, hopefully. But just to kick off, the main raw materials, there are only 3 raw materials which are used in the production of whisky. That is our malt, our water, and our yeast. So only 3 ingredients that go into our whisky production. And the first stage of producing the whisky is to extract the sort of sugars, the starch, fermentable sugars from our raw materials, our barley. And this is done by through a process of malting and then mashing. And the malting process is a process in which we trick the grains, the barley grains, into thinking that they're going to be germinating. So we're tricking them into starting to grow, and therefore produce the enzymes which are needed to break down the energy for the plant growth, which is contained in the starch, which is in the grain. And these enzymes are developed during the malting process. We add water to the grain. And then, trick it into producing these enzymes. Once the enzymes have started developed sort of sufficiently, the malt is then kilned, and this is to stop any sort of breakdown of the starch during that malting stage. And at this point, you could add peat if you were going to produce a peated malt. So this is where you would add peat into the process. It's then moved on to the mashing stage, and this mashing stage that's the point when the starch, which is in the grain, is converted into fermentable sugars, so sugars that the yeast can then use for growth, because the yeast can't use starch as a source of energy. So during the mashing process, these enzymes that are produced during malting break down the starch into fermentable sugars. At this point, we have what's known as wort. So this is a kind of this sugary liquid, which is then cooled down from mashing, which can be up to about 85 degrees centigrade. It's cooled down to around about 20 degrees C, and then yeast is added or pitched into this wort. We then go through a process of fermentation. I'm going to talk a bit more about fermentation in a minute. This is usually done in malt whisky production in wooden washbacks, but the majority of Scotch whisky production plants will actually have stainless steel vessels now. So, and that's a whole other debate, is the influence of whether or not wooden washbacks or stainless steel washbacks are preferable. But generally larger companies, larger distilleries will have stainless steel, but a lot of the companies, some of the larger ones, will still have wooden washbacks. So this goes through the process of fermentation, and at the end of fermentation, you typically have, which is basically a beer, which is around about 8 to 10% alcohol by volume. So it's quite kind of low alcohol at that point. Then in Scotch malt whisky production, we go through a process of distillation, and this is typically a 2-stage process in Scotch whisky production, carried out in copper pot stills. The first distillation is known as a wash distillation, a wash run or stripping run. And this is where the alcohol is basically concentrated up from your 8 to 10% up to around about 25, 30, up to 35% alcohol by volume. The sort of heart, you'll usually cut a proportion of the start called your foreshots, and a portion at the end known as your feint. But the heart of that run was then moved into your spirit still, and your spirit still is used to again further concentrate the alcohol from your 35% ABV up to around about 70% ABV. Typically, that's around about 70% ABV is the final sort of spirit coming off the still in a malt whisky distillation. If you're doing a grain whisky distillation and using continuous stills, you can concentrate that alcohol further, but the maximum permitted distillation is under the Scotch whisky regulations is 94.6% alcohol. That is so that you still leave a proportion of the spirit which contains the flavour products, flavour compounds that come from the raw materials that you've used. So once you've got your spirit that's come off your still, you then need to dilute it down into in order to mature it. So typically it's diluted down from your 70% ABV down to 63.5% alcohol. And this is a kind of a sweet spot, which is known to be quite good for introducing the various characteristics from your barrels. So it's cast at 63.5% ABV, and it has to be matured for a minimum of 3 years under the Scotch whisky regulations. And after which it's then typically diluted to a minimum of 40% ABV when you bottle. So when you're sort of looking at this process, depending on who you ask, you'll get a different answer as to which is the most important stage in terms of developing flavour in your whisky. A lot of the maltsters will say, well, it's all about the malt, so it's all about the barley variety and the malting process, whether or not you're using peat or not. Whereas the distillers will say, oh, well, it's the distillation stage, it's where you make your cut points when you're distilling. And then if the blenders and so on will say, well, it's a maturation. It's definitely the cask that's the most important part of the process. And if you actually Google up, sort of whiskey flavour, you tend to get all these answers as that's where it's all come from. And the main sort of thing that produces all this whiskey is often left behind. But yeast, of course, we need yeast to produce the alcohol. But we also know that they are significant in terms of developing the flavour of the final product too. So that's the focus of today, is looking at the yeast that is used in Scotch whisky production. So on the next slide, we have just to talk about some of the strains that are typically used in the production of Scotch whisky. The main yeast, pretty much all of the distillers in Scotland use very few yeast types in their production, and they're all strains of Saccharomyces cerevisiae. So that's literally translates as sugar fungus, Saccharomyces, and they are produced by yeast companies. So they will buy them proprietary yeast, or buy these yeast from yeast companies. There are three main sort of companies that most distilleries buy their yeast from. There's the Kerry Bioscience, which produce distillers M and MX, AB Biotech, which produce M and MG+, and Lalmand, which produce the Distillmax series. And these yeast are known to the distillers as being consistent in their production. They produce the right yield, and they produce a consistent fermentation, and consistently produce the flavour compounds that are desired in whisky. And these distillers' strains kind of were really sort of developed through the sort of 1960s, 1970s. That was when these yeast companies really kind of took off and became, sort of dominated the scene. So we know what they do and how they behave, and those are the ones that are typically used in Scotch whisky production. The Saccharomyces cerevisiae strains, most of these strains that are used in distilling are diastatic yeast, and that means that they can also break down some of the more sort of complex carbohydrates and use those in their for growth. So they're more efficient, and they're quite high yielding in terms of ethanol. And Saccharomyces species are quite unusual in that they're kind of rare, in that they have absolutely no requirement for oxygen. and they can grow under strict anaerobic conditions. So most animals will die, like us, will die without oxygen. But brewers' yeast don't actually need, these distillers' yeast don't actually need the oxygen at all. And that's what makes them particularly good for production. Other microorganisms within the fermentation could include some bacteria, and particularly lactic acid bacteria, which are considered to introduce flavour compounds that are quite beneficial. During the production of Scotch whisky, the wort that is made is not boiled, unlike in beer production. So there are a range of microbes that come from your raw materials that can get into your fermentation, and actually generally lactic acid bacteria are actually desired in the fermentation process. So on the next slide we have the sort of composition of the wort. This is the environment that the yeast are added into or pitched into. And as you can see from this slide, the predominant sugar that's in the wort is maltose. So 46% of the wort is maltose. So any yeast that you're going to use in production must be able to cope with, must be able to use maltose and maltotriose and so on from this wort, and be able to use those for growth and for fermentation. So the environment that they're put into is quite high in terms of osmolarity. It's got high sugar content. It's usually 10 to 20% sugar in your water, in your wort. And there are also a range of different nitrogen sources, vitamins, minerals, and so on. So the environment is quite specific. It's quite unique, and anything that you're going to use in production must be able to tolerate those the kind of conditions that they're found in whisky production. On the next slide, we have a sort of typical microbiological profile of a distillery fermentation. They're generally quite fast. They happen within 48 hours. You have a very rapid rise in rapid sort of yeast growth over the first 24 hours. And that's when the bulk of your ethanol is produced. So they are quite fast fermentations. And then generally there'll be a proliferation of lactic acid bacteria within the fermenter that sort of kick off around about 48 hours in. And distilleries will vary on how long they actually carry out their fermentation for. Some will keep it quite short. They will stop after 72 hours or so on, whereas others will extend that fermentation beyond 96 hours, perhaps even longer than that, 120, 140 hours. In order to encourage that lactic acid bacterial growth, and then the formation of various flavour compounds that are actually desired. So that fermentation time does vary depending on which distillery is doing it. The temperature of fermentation is not controlled, so it can raise quite high. It starts off around about 20 degrees, and it can go up to as high as 35 degrees C. So they're quite high temperature fermentations compared to brewing fermentations, for example, which are generally carried out around about 18 to 23 degrees C for an ale, and then about 15 degrees C for a lager. So in comparison to a brewing fermentation, the temperature is quite high, and that's quite significant because it means that the yeast strains that you're using in these fermentations must be able to tolerate that high temperature. So the next slide, what's actually going on inside the yeast cell? I would put this one in just because it shows the sort of complexity of the reactions that are going on inside a yeast cell. The Saccharomyces cerevisiae it was the first eukaryotic cell to be genome to be fully sequenced. It was actually sequenced in 1996, and it's a model organism for things like medical research. So we know an awful lot about what's going on inside the yeast cell. But the important thing to sort of that are to remember, and that I usually sort of say to students when they're sort of looking at what's going on inside the yeast cell, is that the yeast that we're pitching into our distillery fermentations, they don't know that they're there to produce whiskey. So they don't know that they're there to produce particular flavour compounds or to produce ethanol. They just want to survive and grow. And it's just a happy coincidence that during the metabolism of the sugars that are in the wort, they produce ethanol, and they also produce a range of different byproducts that also happen to be flavour active and beneficial for our whisky. So the main sort of feature that's important about yeast is, of course, their metabolism. So the next slide. We have the fermentation pathway. Now, as I mentioned, Saccharomyces cerevisiae are capable of growing in the absence of oxygen. Anaerobic respiration, or using oxygen, is the most efficient way of generating energy. But the yeast cells can also adapt a fermentative metabolism. And Saccharomyces cerevisiae exhibit what's called the Crabtree effect. And this is in the presence of glucose, high sugars, basically anything above about 1% glucose in their environment. They will switch to this fermentative metabolism, regardless of whether or not there's oxygen there. So they will ferment the sugars that are available to them, and therefore generate energy. It's not quite as efficient as the aerobic pathway, but it's enough to keep them growing and growing quite actively and well. So in the fermentation pathway, initially the glucose that starts off is converted into a molecule called pyruvate. And during this process, there is a formation of energy in the form of a compound known as adenosine triphosphate, or ATP. And this is known as the energy currency of the cell, and it's used to therefore for growing, for helping the cells to grow, to form more cellular material, and so on. Now, after the pyruvate has been produced, what we're needing to do, the cell needs to do, is to kind of recycle this NADH molecule, which is nicotine adenine NAD. I've kind of forgotten. It needs to recycle this NAD so that it can then break down more glucose and form more ATP. And so the pyruvate is converted into acetaldehyde, and in this reaction, carbon dioxide is formed. And it's this carbon dioxide, this is a reaction in which the carbon dioxide that carbonates your beer or causes your bread to rise, that's where the carbon dioxide is formed in that reaction. The acetaldehyde is then converted into ethanol, and that recycles this NADH for further breakdown of the glucose. So that's the reaction that's sort of central to the metabolism of the yeast cell. But in addition to this reaction, obviously it doesn't occur in isolation, and by happy accident, there are various other byproducts from this central reaction, which form a range of different other compounds. So on the next slide, we have a picture of a yeast cell, and we have this central sugars into glucose, into pyruvate, and into ethanol, have that central reaction. But then we have all these other reactions that come off. There are a range of different products that are produced, such as acetic acid, fatty acids, esters, higher alcohol, aldehydes, keto acids, and hydrogen sulfide. And most of these compounds are quite flavour active. The main products of the yeast metabolism are shown on the next slide. And we have, as you see, the range of these different compounds have a range of different flavour attributes. Now, these compounds are known as congeners. They're basically flavour active compounds that they have a very low flavour threshold, and that's why they're significant. Even though they may be present in parts per million concentrations within your spirit, they are picked up by our senses, by our aroma and taste, and they produce a range of different flavours and aromas. So our higher alcohols produce things like alcoholic and fruity, banana, fusel, or pharmacy type aromas and flavours. The esters are the ones that are the main ones that produce the sort of fruity apple, banana, creamy, pineapple kind of type fruity type notes. And then we have acetaldehydes, aldehydes such as acetaldehyde, which has a sort of green apple type flavour, and diacetyl, which has a sort of butter or butterscotch type flavour. Now, if we have a focus on the esters, which are the most significant in terms of sort of new make spirit or unmatured spirit flavour, we know that various things about how esters are produced within the yeast cell, and the next slide sort of shows this. So these esters are produced through this sort of ester synthase pathway, and they're impacted by the oxygen that's available in the environment, unsaturated fatty acids, and also things like the fermentable sugars that are available in the wort to start off with, and the nitrogen compounds that are available too. So if we look on the next slide, we can see the parameters that actually help us, that actually impact on the production of these fruity flavour compounds. If we have a high original gravity, so a high sugar level at the start there, the higher the gravity, starting gravity, the higher the ester production. Similarly, if you have a high number of free amino nitrogen compounds, so these are things like amino acids and peptides and so on, again, they cause an increase in ester production. Things like lipids, which can be produced through processing through the mash filter or so on, they will cause a decrease in the amount of esters that are produced. And similarly, so will aeration. So if you have too much oxygen present, again, that will decrease the esters formation. Other factors that impact on ester production include temperature. So the higher the temperature, the more esters are produced. And the pitching rate of the yeast, so how much yeast we add in at the start of fermentation. If we add sort of too much, it can have a negative impact on our production of esters. So we know that we can carry out certain things in terms of our processing that will impact on the production of flavour compounds. But there are other ways that we can influence the amount of flavour or the types of flavours, the flavour compounds that are produced. And that is by changing actually the yeast strain that we're using. So on the next slide, just to highlight under the Scotch Whisky Act, the only thing that relates to the microbiology of the production process is a statement that Scotch whisky must be fermented at a Scottish distillery only by the addition of yeast, but it doesn't specify what yeast. So it doesn't say it has to be Saccharomyces cerevisiae. So this sort of statement gives us allows us a bit of freedom to try and investigate other yeast strains or investigate playing around with our yeast to try and generate different types of flavour. And there are different approaches that we can take to looking at this. And the next slide, we carried out some work with Dornoch Distillery recently, looking at the various yeast strains that we obtained from a culture collection. And these culture collection, we separately identified these yeast based on their ability to use maltose as a sugar source. And looking at different yeast strains, in this piece of work, we just carried out the fermentation point and then assessed the wort, the fermented the wash at the end of fermentation. So the only difference between each of these different washes was the yeast strain that was used to produce it. And we found that there was quite good variation in the sort of flavour notes that were picked up from these different types of different yeast strains. And these varied from Saccharomyces to Torulaspora, Brettanomyces, a range of different other yeast species. There were notes such as tropical fruit, red fruit, strawberry jam, clementine, apricots, raspberries, all sorts of plums, all sorts of different flavours that we didn't get from a Saccharomyces sort of general strain that was used typically in the whisky production. Another approach that we could use is looking at commercial strains that are already available, but used in other spirits production or in the production of wine. So on the next slide, we have some results from a project that we've just finished with this Port of Leith distillery, looking at different strains that have been used in rum production, in wine, and various types of beer. And we assessed 23 different strains for their to look at to examine the flavour compounds that were produced by these different strains. And we carried out this test known as a main acceptability rating. It was a hedonic scale, a 9-point hedonic scale. And this sort of scale this type of testing is used to test sort of things like food products and things for acceptability in terms of sensory quality. And we found out of our 23 strains that some of the ale strains, the English ale strains, for example, like Kvik ale, which is a Norwegian yeast strain, some of these Belgian strains and German ales and so on, all produce spirit that was equal to or around about the sort of same acceptability rating as our control, which was the distillers' strains. And all these strains, they were able to produce a yield that was commercially viable, because that's an important part of the piece, that you need to make sure that you're producing enough ethanol that it makes the process commercially viable. But they also produced a range of different types of flavours and so on, which were regarded as being highly acceptable in terms of their sensory quality. So we can look at other commercial yeast strains as well as strains that we can get from culture collections. The other way that we can investigate other strains is to actually carry out some environmental sampling and try and isolate some wild yeast from the environment and assess those for their ability or suitability to be used in whisky production. So the next slide leads us on to Lindores Abbey. Now Lindores Abbey is based in Newborough in Fife, and it was founded in 1178, the actual original abbey. And there were various sort of outbuildings and things that kind of expanded over the years as the number of monks grew into quite a large area, and they developed a distillery on the site. And Lindores Abbey is regarded as being the birthplace of Scotch whisky, and the reason for this is that there was a note in a transaction which was recorded in the Exchequer records in 1494, which stated that to Friar John Core, by order of the king, to make aquavitae, 8 bowls of malt. And this was the first recorded transaction of malt to be used specifically for the production of aquavitae or early whisky. So this is why it's regarded as the birthplace of Scotch whisky. And we were contacted by the owner of the Abbey in kind of 2012 when they were beginning to look at establishing a new distillery on the site. Happily, that distillery is now up and running. It's an absolutely amazing place. So if anybody ever gets a chance to go and visit, please do go and see it. And they've released their first whisky, so it's up and running and going again. And the reason why we were contacted was to have a look at the sort of the flora of the environment around the distillery, to see if we could isolate some yeast species from around the distillery. The idea wasn't to try and recreate the original whiskies that were produced, because we know that the environment's completely changed, the plants, everything was changed, so we wouldn't be able to isolate the same kind of species. But the idea was that we could isolate some yeast that could potentially be used in production as a good story from for the distillery. Because of course the initial sort of fermentations that would have been carried out in 500 years ago, it would have been yeast. It would have been probably open to the environment. It would have been left to the wills of the gods as to which species proliferated and grew in the fermentation. So it was likely to be a kind of wild type fermentation that would have been the initial production of the aquavitae. So what we did on the next slide, we had a sort of a scheme for looking at the flora around the distillery. We carried out some sampling using different methods, and we screened using media that contained ethanol, because we wanted to make sure that we isolated yeast that were capable of tolerating alcohol, because they had to be able to tolerate it and be able to produce ethanol. So we carried out this screen. We looked at their potential to grow at temperatures as well, at 37 degrees C and at 29 degrees C. We carried out fermentation tests, and we carried out identification as well of a range of different organisms, and we did some sequencing so that we could identify some of the species that we isolated. So on the next slide, we have some of the sort of pictures of the sampling that went on. On the sort of top right, we have an air sampler. This is sorry, the top left. That sort of machine that looks a bit like a lighthouse is an air sampler, and that is basically like a vacuum cleaner. Actually, you kind of put a petri dish with your media in the top, and you basically draw over air from the air over the top, over the plate, and that sort of helps inoculate the plate. We also did things like we left plates open to the air. We left them in sites that we considered to be nearest to the building that was likely to have been the original distillery. And we also kind of did swab samples as well. So we carried out swabbing samples of around the abbey too. And we isolated hundreds of colonies, but we did sort of, we screened them down, and we tested them for their ability to actually tolerate different strengths of alcohol and to ferment or to grow at particular temperatures as well. And carried out some microscopy to try and identify those, have a sort of initial sort of screening to make sure that it was yeast that we were isolating and not bacteria, for example. Next thing we did on the next slide, we have some fermentation trials, and this was we took sort of the top 5 sort of candidates from our screening and put them in, carried out a sort of a fermentation test to have a look at how it compared to a traditional distilling yeast. And as you can see on this slide, we have our M yeast, which is our distiller's yeast, as our control, which attenuated very well. So the attenuation is basically your drop in gravity going from your high sugars, that's consumption of sugars, through over up to your sort of 48 hours. You've got that rapid consumption of your sugar and the production of ethanol. So the control worked, but as you can see, all of the strains that we isolated had very different fermentation profile. Although the majority of them did actually come to the same point at the end, it was after 140 hours. It was after 120, 140 hours before it actually reached the same point as the control sample. So if you're looking at using this in a commercial distillery, it's not very efficient. You'd have to leave it fermenting for significantly longer period than your typical distilling yeast. So although most of the sort of flavours that we sort of got out of this were kind of more fruity, there were some that were quite estery, these sort of fruity and yeasty notes that we had from these. But it would be a balance for Lindores to actually decide whether or not we're going to actually use these in production, because you've really got to think of the equipment and your space and your yield and so on, and whether or not that these addition of these different flavours actually made commercial sense. So there's still a bit of work to do on this, but we'll be taking this forward with Lindores and working more on this as we go. So just on the next couple of slides, when I've sort of do a talk, I generally talk about yeast quite a lot, but when I'm doing this, I have some samples from a project that we did with the Oxford Artisan Distillery. And in this sample, they were trying to do the same thing that we did with Lindores, and they sampled the barley fields that were outside the distillery, which were the ones that were going to use in their, for their malt. And they carried out a range of different sampling of the environment, so lots of plants and things. But they also isolated some yeast from the distillery cat, which was one of these hairless cats, and they also sampled the mustache of the head distiller, which is Corey Mason. So Corey Mason has got this. He's one of our graduates, and he's one of these. He was an bartender in the States, in New York City. He's got one of these handlebar moustaches, and they isolated some yeast from his moustache. And what I usually did, what we found in the project work that we did, and what we've found when we've given out samples to a lay audience, is that actually as we see on the next slide, the most favoured sort of spirits, the best sort of characteristics we got in terms of the spirit that was produced were from the mustache and the cat. So things like we got these fresh fruits, floral fruits, tropical notes, and so on. And so when we do these sort of taste tests, we find that the yeast from these from there actually produce very good, very positive flavours. So it's just a way that a lot of distilleries are sort of looking at using different yeasts to see if we can impart novel flavours or a wider palette of flavours to the spirit that they are producing. On the next slide, just to mention, we're not the first people to look at this, to look at yeast and the alternatives and what they can produce. There was the Saccharomyces diamath, which was isolated from the barley fields outside Glenmorangie. That was developed by Bill Lumsden. He developed this over a long period of time, and they released their Alta, which contained this. It was the spirit that was produced from this novel yeast. They released that in 2019. And there was also a release from Glenelgin in 2017, in which they used a yeast strain called Schizosaccharomyces pombe, which produced a kind of creamy note. So there are companies that are already using alternative yeast to these traditional, these proprietary yeast that are produced by our yeast companies. In the US, actually, a lot of companies have proprietary yeast, and they guard them, and those are the ones that they use for their production. So it's quite interesting to see that we're kind of doing this sort of thing as well, and companies are looking at producing or having their own yeast strains which are bespoke to their company. And a number of the yeast companies, such as Lallemand and AB Biotech, are actually looking at working. They're actually working with distilleries to produce bespoke strains which they can then use in their production. So finally, I'd just like to thank all of the students that have been involved in the projects that we've carried out, to thank the companies that we've been involved in, so Dornoch, Portofleet, the Oxford Artisan Distillery, and of course Lindores Abbey. And finally, to thank the IBD for sponsoring the talk, and thank you for listening. Are there any questions?
Oh my goodness, what's happened to my background? There we go. Annie, God, that was just superb. You brought me back to my R&D day, which I have to say was a long time ago, but thank you so much for that. The word you were desperately trying to get out was nicotinamide adenine dinucleotide. Brave person to attempt to do that in a talk. But thank you very much for that. Try. I love the end. I love the last few slides. The cat whiskey, great. And that was a picture of a very young Bill Lumsden in that Glenmorangie. Yes. So we know each other very, very well. Yeah. I'm hoping the tech team can maybe give me an extra 5 or 6 minutes because there's a few questions have come in actual fact. See the distilleries which were using very long fermentations. I think you mentioned your graph went to 96 hours, but you said it could be up to 120 hours. Mm-hmm. I mean, that must be sacrificing like throughput and total alcohol production. I mean, that's an admirable thing for a company to do.
Yeah.
To make quality a priority, basically, Annie.
Mm-hmm. Yeah, no, definitely, and it is finding that balance. There are some companies that will do their quick run over, say, like Monday and Tuesday or whatever, and then they'll set off the next fermentation, and then leave it over the weekend. So there are some that will do it that way, and they'll have a mixture of short and long fermentations to try and sort of increase, make sure that they're sort of maximizing their yield, but and then they'll blend the two together. So there are some that will do that. But yeah, it varies depending on which distillery you're looking at. A lot of the larger distilleries will be all about yield and and pushing it through. And they produce, I mean, the strains that are available that they use in commercial production, they do what they want them to do. They produce the flavour compounds that they want, and if it's not broke, don't fix it.
Yeah.
So it's not an approach that's going to be widely used, but it's I think with the advent of craft distilling and the entry of the sort of smaller companies, they've got more opportunity to do this, to do it on a small scale, because it's a lifestyle choice rather than a trying to make money kind of thing. With a whiskey, so that they will be more experimental, and they will they'll have the opportunities to sort of do this and use it as their USP, because they're trying to compete against these big boys. So you've got to try and have something that that makes your spirit unique.
Yeah. Now you mentioned the use of Schizosaccharomyces pombe. Now I remember those trials at Glenelgin and also at Oban. And Oban 1 came out, the new make spirit came out with a very distinct blackcurrant type flavour. So there's an interesting thing, trying an experiment with and you could get different flavours from different distilleries. Now, the distinct flavour you're talking about is in the new makes, well, certainly was in the new make spirit when I remember we identified it. Does that very distinct flavour, does that manage to survive, to shine through after maturation, Annie?
Well, that's the thing, but because there are so many other factors again that will impact on the final complexity of the spirit, depending on how long you mature for, and what kind of casks you're going to be using. And so there are all sorts of other aspects that will impact on just how much wood character is going to come through, or the maturation characteristics that are going to come through from your from the maturation point. But and then we've not actually, one of the things we haven't done and we're planning on doing is doing some accelerated maturation trials to see that, see whether or not it does actually convert to well, hopefully a more sort of balanced flavour profile, and just to determine exactly what, how much of this character does follow through. But we do know that the Ester character does still convert through, still maintained within the spirit, no matter how long it's matured for. So
yeah. Now I know that the nosing and the taste flavour chemicals, it's very rarely due to one single chemical. It's a whole balance. And there's Robert Bonates asked on YouTube, I recently read that chemists have found more than 10,000 chemicals in beer. How many do you think there are in whiskey?
Well, it's a similar sort of level, isn't it? It's the same thing as Saccharomyces or yeast that are producing these compounds, and of course you've got all those characteristics that you'll find in beer, apart from the ones with hops, apart from the hop characteristics, and then added on those maturation characteristics, all your lactones and everything that comes from your vanillin and everything that comes from your wood extraction. So yeah, I mean, we know that that there are thousands and thousands of compounds, and that's one thing that I've been working on quite recently is looking at that sort of chemical footprint, and whether or not you can identify or try and predict what kind of footprint you're going to get from your yeast in your final analytical analysis of your product. But one of the difficulties with looking at these all the different compounds in a whiskey, the way that they're detected and reported differs, and there are different techniques. And so most of the sort of main techniques will pick up a certain type of compound or certain range of compounds. There's no one technique that picks up everything that says that this is your actual breakdown of your spirit. And what your analytical chemistry also doesn't tell you is what the interactions, the synergistic reactions, the balance of compounds that you'll get. So you might have a chemical footprint, but actually it will taste very different to what it looks like, what the chemistry looks like. So there's an awful lot still to do, still to learn, which is good for us.
Yeah. Now you mentioned there was people are experimenting with different yeast types and strains and things. Now I saw one of your slides, there's basically 3 companies that provide the yeast to the whole of the distilling industry, and I know the yeasts are fairly similar and they produce fairly similar yields and flavour components. Now I also know that there are very high alcohol yielding yeasts available. And these yeast, they can produce anything between 16 to 20% alcohol yield. And I know, again, you said it's about 8 to 9% in the fermented wash in a current distillery.
Yeah.
Has anybody tried those extremely high yielding yeast?
Well, because there's also been increasing interest in looking at high gravity fermentation, so increasing your sugar content at the start, a bit like in brewing, you use high gravity fermentations and then you dilute it down to produce your final beer concentration. But yeah, there are companies that are looking at these, and I get, but again, it's getting that balance between your yield and your congener production. That you need to get that balance right, and as we've sort of found with only a few companies supplying the majority of distilleries in Scotland with their yeast, if it's not broke, don't fix it. It's like kind of a lot of it is to do with not wanting to make any changes that could actually be detrimental to the process.
Yeah, you also early on you showed the traditional fermentation vessels, the wooden washbacks. And I know the industry has been migrating over to stainless steel, and there was always a huge debate, and I'm sure there is still a debate going on. One of the debates in my day, which is not that long ago, was that wooden washbacks, if you didn't sterilize them often enough and properly enough, you could actually introduce contaminants. You could introduce flavors like butyric and things, which both affected putting flavors that you couldn't get rid of, that you didn't want, and also it reduced the yield. Is that still the case? Yeah, that's
I mean, generally they're sort of steam sterilized, aren't they? I mean, generally they're quite they're quite well treated, so they there aren't as many incidences of these sort of wild fermentations that cause really detrimental issues, but yeah, there's definitely, I mean, that's one of the projects I've got at the moment, actually, is that we've actually got some a part of an old a washback from a distillery and that we've actually done a CT scan on and seen that there's various there's obviously something there that's on the wood, on the surface of the wood, and within the first few layers of the wood that is obviously maintained even when it's the washbacks have been sterilized or steam sterilized. So we know that there are there's a microbial flora that's unique to every washback, and that's one of the things that we're looking at. We're looking at the production of actually specific methyl ketones and their influence on spirit flavor as well. But those are regarded as being a positive flavor rather than a negative one. So there's pros and cons for having those microbes there.
I've got two more questions for you. In one of the I think almost the first slide or second slide, you showed a picture of the IBD, the labs and the pilot plant. See this, the alembics and the 5 litre plus stills. How representative of the distilling efficiency and of the pneumic spirit quality comes out of that? How representative is that of full scale malt distilling production?
Yeah. We have actually, because that was one of the port of Leith in our sort of initial sort of preparations and trying to get our methods kind of right. That was what we looked at, whether or not we got basically the same kind of spirit at 1 litre, 2 litre, as you would get on a larger production. So we have actually looked at that in the spirit that we've the sort of analysis that we've done, the nosing and whatever. The spirit is pretty much spot on with a sort of larger production. So we're kind of pretty confident that even though it's done on quite a small scale, that it's representative of a much larger production run. So yeah, the actual distillation part itself is we're kind of quite confident that we've got that right.
Okay, and the final question for Yanni, what do you think will be the main developments happening in malt distilling in the next decade?
Well, one of the main things, of course, one of our we've got very ambitious targets in terms of meeting our environmental requirements, like the environmental needs. And so obviously the industry, I mean, as a whole, it's been very proactive in looking at reducing energy, reducing water use, and so on. So a lot of the focus is on reducing our energy consumption, finding alternatives to our oil, fossil fuels and so on for producing our spirits, and also reducing the amount of water that we're using in the process. It's a very water intensive process as well. So looking at methods of reducing those without impacting on the quality. So it's maintaining the quality of the spirit, because we know we've got an amazing product, and so it's meeting all these targets and doing it in a manner that is sustainable, so that we can carry on this pass it on for generations to come. Similarly with things like looking at sourcing our raw materials, our barley, for example. We know the impacts of climate change are significant already, and that these will only be increased. So looking at maintaining the quality of our raw materials as well. So we're maintaining our barley, barley quality, being able to grow it efficiently, and being able to keep our supply going of our barley strains as well. So I think a lot of the focus will be on the kind of environmental side, the sustainability side, and making a way of future proofing our the product, because we know it's special.
That's wonderful. There is plenty to keep you occupied, and plenty to keep people in employment. And as long as you keep making that lovely golden nectar, Annie, I'll be a happy man. Now, on that note, I'd like to thank our audience for taking part and submitting their questions. And could I just ask you to complete a feedback form for us here in the festival? Don't just need to do it just now, but there's a link to in the YouTube description below. Thanks for doing that, because it helps us so much in shaping things for the future. And of course, Annie, thank you so much for that fascinating and thought-provoking talk. It's certainly going to ferment thoughts in our heads, and hopefully we'll distill some sense out of them. Now, the following and final event in the programme, the closing ceilidh. It's in the Festival Club at 8:30 p.m, so just 5 minutes or so. Everyone is welcome. It's great fun. If last year was anything to go by, it certainly is great fun, and you're all most welcome to come along. The best thing to do is head straight over there once I've finished here. If you've enjoyed the festival, you can consider donating. The details are in the description box. And don't forget to like us on Facebook, Twitter, and Instagram, and follow this YouTube channel, because you can keep up with all the things that go on throughout the year out with the major festival event. So thank you all so much, Annie. Thank you once again. That's it for this year. Goodbye for now.